Bismillahirrahmanirrahim. Synthesis, storage, and release of serotonin in serotonergic neurons. Uh, Consider this is the neuronal cell. From the food, L-tryptophan is get absorbed by carrier transport mechanism, and this L-leave form of tryptophan amino acid. This then get entry into the cell. Inside the cell, hydrolase enzyme converting L-tryptophan into 5-HTP, 5-hydroxytryptophan, and then decarboxylase converting this 5-hydroxytryptophan into 5-hydroxytryptamine, which is known as serotonin. But this 5-HT is then get metabolized by monoamine oxidase enzyme, dehydrogenase enzyme, and so on. Once the 5-HT is formed in the cytoplasm, this 5-HT is then transported into the monoamine vesicle and stored here. The question is that why these neurotransmitter autocoids are not free here, but they are stored in the vesicle. The answer number one is in the cytoplasm, 5-HT is get metabolized and inactivated, but inside the vesicle, 5-HT is not metabolized. Second one, if the 5-HT is scattered in this cytoplasm, there is no release mechanism for individual 5-HT in this synaptic space to react with cells. Number third answer, the neurotransmitters are released due to increased calcium level is exocytosis in pockets, and there is release of neurotransmitter in bulk amount in this synapse, and then these neurotransmitter react with its respective receptors. Now, once they are form vesicle, inside the vesicle, amino acid, ATP, other proteins are packed. When action potential coming here, sodium is get internalized, sodium changing the electric concentration of this cell. This change electric concentration of this cytoplasm, opening the potential dependent calcium channels, and now calcium influx occur. This more calcium level accelerating the exocytosis of these neurotransmitters. Once the neurotransmitters are released, now they are get metabolized by the various type of enzymes in this synapse, in the synaptic cleft. They can reuptake by this transport mechanism again into the cytoplasm, and again they get stored here, and they react with its respective receptors. This is 5-HT1, 5-HT2, 3, 4, 5, 6, 7, 6, 7. But there are some drugs like PCAPA, parachlorofenylalanine, which is also known as phenylalanine. This is the hydrolase inhibitor. Once there is inhibition of hydrolase, the synthesis of 5-HT is blocked. This PCPA was first time used to treat carcinoid syndrome, but due to its adverse effect, because this PCPA completely depleted the concentration of serotonin, and this completely demolishing the concentration of 5-HT3 was very toxic. So further development of this drug was not carried out. Similarly, this decarboxylase enzyme is blocked by PC parachloroamphetamine, and this is neurotoxic. That's why this drug was also not developed. This monoamine oxidase enzyme is blocked by monoamine oxidase inhibitor drugs, and this is the side effect of these monoamine oxidase inhibitor drugs. A side effect. And this reuptake is blocked by CSSRIs, selective serotonin reuptake inhibitors, TCA, tricyclic antidepressant, and cocaine. Cocaine and these blocking these reuptakes.